Okay, hi there. Welcome to another episode of Science and Technology, History of Science and Technology Q&A. This is on a different day than usual. My apologies for switching things around. This is a time of year where we're in the middle of our summer educational programs, and I keep on getting pulled into things related to that. In fact, we'll be live streaming a few of the events probably from our summer school. We have well, 3 or 4, depending on how you count it, different programs all running. Right now, this week, we have a middle school summer camp. Next week, we have week 0 of our summer school. That is the pre-week for the physics track of our summer school. Then we have 3 weeks of our summer school. And in the last 2 weeks of that 3-week period, we also have our high school summer camp. So anyway, lots of educational programs. The middle school one is a little easier for me because we are not assigning individual projects to everybody, and that's part of my responsibility for our summer school and high school summer camp. So that's kind of my moment of extreme professoring in the year when we'll have, I don't know, 100 and something students, and the goal and the thing we always achieve is to come up with unique original projects for each student. And I typically make a list of potential projects, and people pick partly from that list and partly from things they want to do. And then I'm kind of responsible for the final selection there. Anyway, lots of fun for me, but also rather an intense period with rather tight scheduling. So these live streams might bounce around a bit, but there may be some interesting, unique additional live streams, which we'll be doing as part of particularly the summer school. All right, so let's see. There were all kinds of questions left over here. There's one from Mikhail about how was DNA discovered, history behind its discovery. Not sure I can tell you all of the details there. A few of the characters involved I do know somewhat. I mean, Jim Watson is still alive. I've certainly met a number of times. And Francis Crick, I knew somewhat. Those were the sort of 2 most credited people. And I guess Jim Watson wrote this book called The Double Helix, which is kind of a adventure storybook about the discovery of DNA in 1953. I think its first sentence is, "I have never seen Francis Crick in a modest mood," or some such other thing. I'm not sure that would quite be the way that I would describe at least my interactions with Francis Crick, but of course I knew him much later in his life. There's a little bit of the science of what was going on. I mean, before, people knew that there was DNA in the cell nucleus, and that had been known for a long time. I think people back from the 1920s, 1930s had known that. I don't know how much Thomas Hunt Morgan, who was a big sort of fruit fly genetics person, I don't know exactly how much he knew. I think he knew things about the molecular, what kinds of substances were involved in the cell nucleus, but just how heredity worked wasn't. I mean, it was certain laws of heredity were known. They'd been figured out by Gregor Mendel, and other people had figured out these, heritable traits and these different kinds of dominant recessive traits, all this kind of thing. But the mechanism by which that worked was not clear. And there was this sort of idea of genes and so on, which were all a bit kind of based on the observation of what effects they had rather than the mechanism by which they were made. And so there was DNA, it was in the cell nucleus. It probably had something to do with heredity, but it wasn't really known how that worked. And there had been some vague thinking, I think, that it might be something that was a little bit like kind of storing data or something. But the idea of storing data wasn't yet a big thing, because remember, historically, 1953, which was sort of the big year for the figuring out of how DNA works, that was something that was in the very early days of computers. I mean, today, when you say there's information, there's data, it's stored digitally, everybody would know what that meant, because everybody is used to kind of the world of computers, but that simply didn't exist in those days. The typical, let's say, physicist or something would never have seen a computer. The typical biologist would never have seen a computer. I suppose there was so this idea that there would be this sort of digital information thing wasn't really a thing yet. I'm kind of thinking that, and I may have my history slightly mixed up. There was a physicist named George Gamow who was also much involved in the origination of the Big Bang model of the early universe and so on, who maybe it was after the actual main discovery of DNA, but he was involved in later in figuring out the code by which you go from base pairs to proteins, to amino acids to proteins and so on. But I think that there have been some suggestions that maybe there was some sort of digital information component to DNA. Okay, but then the real question was, there's this molecule. What's the structure of the molecule? So a few other molecules, biological molecules, their structure had been figured out. Normally, how do you figure out the structure of a molecule? Well, the big method that was used was X-ray crystallography. So the thing to understand is, in a crystal, the atoms are arranged in a very regular periodic way. The question is, in what way are they arranged? You have a diamond crystal, you have a salt crystal, the atoms are arranged differently. In a salt crystal, it's a cubic arrangement of atoms. In a diamond crystal, well, it's usually called the diamond arrangement. I guess it's a hexagonal-ish arrangement of atoms. And the question is, what is the arrangement of atoms? And there's this technique called X-ray crystallography, which is a way, given that you have a crystal, it's a way of saying what is the arrangement of atoms in the crystal. And it works by reflecting X-rays from the crystal. And when the X-rays are reflected specifically from the atoms in the crystal, and depending on how the atoms are lined up, you get lines of destructive or not, or constructive interference between the emission of the re-emission of X-rays from the surface. It's actually very much like the way that a phased array radio works. When in 5G, for example, one has this whole idea of, one will emit radio from this sort of distributed antenna, and it will be all directed in a certain direction because the phases of the different things coming from those different parts of the antenna will be arranged so that the radio energy goes in a particular direction. It's sort of the same kind of idea with X-ray crystallography, except that there's sort of a natural array that is made up by the atoms in the crystal lattice. And so that's kind of the way that one tries to figure out how a molecule is arranged, how the atoms in a molecule are arranged is, well, if you can make a crystal of that molecule with that molecule, then everything is lined up, many copies of it are lined up, and then you can use X-ray crystallography to try and find the arrangement of atoms. Now, one of the big challenges there is how to actually do the crystallization, how to take this material and have it form into a big crystal. And that's a very tricky thing, and the sort of theory of crystallization is still quite incomplete, and it's still kind of often something of a mysterious art to make a big, perfect crystal. It's something very important in the semiconductor manufacturing, making these big crystals, big, as perfect as possible crystals of silicon. But back in the day, people were making crystals of some of these biological molecules. I think hemoglobin was an early one. I think myoglobin was another one. I seem to remember the chap who was, I think it was a person called John Kendrew, who was the person who decoded, I think, myoglobin by crystallizing it and then using X-ray crystallography. Strangely enough, in the weird connections of this world, he was an alumnus of the elementary school that I went to in Oxford, England. And I remember there was a grand sort of science exhibition opening thing when I was like 8 years old, that this then very ancient seeming person showed up to, and I at least said hello, so to speak. I don't know. That was the that shows the difference in timing. So that was that will have been 1968 or so. The person who had sort of decoded myoglobin was by that point an older person. In any case, the big challenge in all these things was how do you crystallize these complicated molecules? Because if it's a complicated molecule, it's flapping around all over the place. It's not easy to make sure that it can sort of line itself up perfectly. But that was kind of a big challenge. I don't know the full history of this. I think Rosalind Franklin in London was the person who was most deeply involved in kind of the crystallization and X-ray crystallography of DNA. But the question was, when you do X-ray crystallography, what you get is a bunch of bright spots from what were the angles at which the X-rays were kind of emitted from the crystal. And then the question is, that gives you essentially a Fourier transform of the actual distribution of atoms. In other words, what it's telling you is, if you look at if there was a periodic array of atoms with a definite period, that would be like a wave of a definite frequency, and that would be like a single spot. But what you're seeing is what arrangement of different periodicities of atoms are there going in different directions, and that's the arrangement of spots that you get. And so there's then a big decoding problem. If you've got this arrangement of spots, now there's sort of the puzzle of, well, what's the actual arrangement of atoms that gives that particular arrangement of spots? And in general, it's not unique. In general, for any arrangement of spots, and technically it's because you only have the intensity, you don't have the phase information of the X-rays, you only have the intensity of the spot in a certain angle. But the question is, given the intensity of spots, given this distribution of spots, what is the actual structure of the molecule that produces that? And as I say, it's not unique. So there could be many different shapes of molecules that could produce the same distribution of spots. Of course, many of those shapes of molecules would not be plausibly constructible out of atoms. And so then there's this kind of almost mathematical problem of, can you kind of reverse engineer the thing to figure out what structure would lead to this pattern of X-ray crystallography output? And I think the dynamics of what happened is, I think Rosalind Franklin was the person who got the X-ray crystallography done. And I think then Jim Watson and Francis Crick were the people, they were working in Cambridge, England. They were the people who figured out kind of what would be the distribution of atoms that would lead to that X-ray crystallography pattern. And they famously had this thing made out of Meccano that was kind of this helical structure that was the thing that they figured out would lead to that pattern of spots. And so that was sort of the big thing, was figuring out that structure. And then the question was, well, so how does that structure actually work? What does it do? And that took a number more years. And I think in the original paper that they wrote in Nature magazine in 1953, they had sort of a couple of sentences at the end saying, this might suggest that perhaps there's a whole sort of digital information story associated with DNA, which I think kind of implies that they weren't the ones who originated that digital information story, but nobody could kind of see how to make it work before it was known what the structure of the DNA molecule was. Now, I think it's sort of interesting to see the kind of career tracks. I think Francis Crick had been doing physics before kind of going into this sort of biology direction. I mean, in his later life, he kind of changed direction and worked on things about the brain and neuroscience and consciousness and so on. I met him when he was in that sort of phase of life. Jim Watson continued very much in the molecular biology, was a co-author of a sort of standard textbook, was very much involved with Cold Spring Harbor, which is kind of it's a place with a very unfortunate history that goes back to the world of eugenics in the early 1900s in the US, but is a distinguished place where a lot of important research in molecular biology has been done. He was also involved, Jim Watson was also involved in kind of the pushing for a government-sponsored human genome project and so on. He's also a person who I would say believes that perhaps the things that he can deduce about the world from science are definitively the way the world has to be, and sometimes I think perhaps downgrades sort of other views about the world and comes to all kinds of conclusions that he says, look, it's scientifically provable that this is true, but actually he's sort of missing a big piece of the picture. But that's kind of his subsequent life. I mean, the thing for which he is best known is the 1953 discovery of the structure of DNA. So that's kind of the story there. And of course, it took a number of years. It wasn't, well, I see the history from far after it happened, but by the 1960s, things were really hopping in terms of the development of molecular biology. As a field, molecular biology kind of started popping up as departments in universities only considerably later, 70s, 80s, and so on, but as something where people were sort of figuring out the key ideas, figuring out messenger RNA, transfer RNA, how that whole sort of mechanism worked. That was really a story of the 1960s, centered around places like Cambridge and Cold Spring Harbor and places like that. And I guess Jim Watson was at Harvard for a while, so it was kind of the two Cambridges and other places. I think one person who was a figure in that whole thing was a person called Sidney Brenner, who I knew reasonably well, who was, for example, the discoverer of messenger RNA, which is, of course, famous now because it's been used in a couple of these vaccines. The Pfizer and Moderna vaccines use that. One of the things sort of interesting about him was, he had been trained as a biologist, but he had a deep interest in computation and sort of computational kinds of things from right when he grew up in South Africa, right when he was sort of first going through school. And famously, he had, written letters to Alan Turing when in the early 1950s, when Alan Turing was sort of thinking about biological kinds of things. But Sidney Brenner was definitely somebody who sort of thought about biology in computational terms. And he encouraged me for many years to take all the stuff that I figured out about computation, how it works, and apply it to biology. Finally, maybe I have some ideas about how to take the next step in doing that application. But the main thing is that for Sidney Brenner, it was kind of thinking about the mechanisms of biology computationally was sort of what came first. It wasn't like, let me go and understand these molecules and then deduce that they are computational. For him, it was very much, I think, that he was thinking in computational terms and trying to fit sort of these mechanisms that he was thinking about computationally into what biological organisms and biological systems actually do. Anyway, so a little bit on discovery of DNA. Let's see. Boy, there's a question here from a Tory about history of musical digital instruments, like synths and computational music and so on. Well, you guys are really testing my memory here. My gosh, I should see. This is so. Let me tell you a few things that I know about that. I mean, the idea of non-standard musical instruments was something that I think in the early 1900s was as sort of electricity became a more common and popular thing. People are like, how can you apply electricity-like ideas to the generation of music? And so, for example, there's this instrument called a theremin that was sort of an early electrically inspired and developed instrument. And I think people like Nikola Tesla were involved in sort of theremin-like things and making the kind of woo noises that theremins make. I think that for a while, these were sort of analog processes where you're just using, inductors, capacitors, resistors in electrical systems. And I think to some extent, I think in those systems, you actually sort of like put your hands in the electric field and you are distorting the field lines of the electric field by where you put your hands. And that's kind of how you play the instrument. And that what you're doing is you're essentially dealing with in an electric circuit, you can have between an inductor, loop of wire, and a capacitor, something that's storing charge. You can make with an inductor and a capacitor, you can make an oscillator. And essentially what you're doing in something like a theremin is you're changing the characteristics of the inductor and capacitor by moving your hands around and moving other elements around. And that is changing the frequency of the oscillator. And so that will, in a sort of a standard note on a musical instrument has a fundamental frequency that is a definite frequency. Let's say it's middle C, it might be roughly 256 cycles per second. And that means it's a sine wave of sound pressure. At least that's the fundamental part of the note. On a piano, there'll be a whole sort of structure of overtones to that note, double the frequency, et cetera, et cetera, et cetera, and other sort of things that are around, that have that as the base frequency, but then there's a whole elaborate collection of other frequencies that are also involved that give the kind of the color, the timbre of the note that's being produced. So it isn't just a pure annoying sort of pure tone, but gives sort of a depth to the sounds. But so the early thing, I think 1920s, 1930s, it was pretty common to see these sort of electrically based instruments that were essentially tweaking the idea of an inductor, capacitor, oscillator type circuit. I think the early kind of more portable sort of synthesizer instruments, I think was his name, Robert Moog, maybe, of the Moog synthesizer company and device. I think, and now I think that is still an analog device. So still something where you actually have circuit elements and you're changing capacitances and things like that. The big moment of producing digital, so the real question is, it's easy to make a piece of electronics that will make a tone of a fixed frequency, and you can have an oscillator in digital electronics. You will typically have oscillators that drive the clock that is specifying, you know, which instruction, the sequence of instructions you use, and so on. But to get a pure tone, a single sine wave is something you rather easily get from an oscillator. The big question is, how do you get kind of a realistic sounding, you know, piano, trumpet, whatever else kind of sound? How do you synthesize that sound? And so the first big kind of innovation in that area was a thing called FM synthesis. And that was in the early 1970s, I think, developed particularly around Stanford. And I am forgetting the name of the chap who I have certainly met, who did that. But the idea there is, usually you have a pure tone as a sine wave. So it's sine 2 pi frequency times time. And that's the amplitude of the sound wave, the pressure in the air is, you know, basically omega times the frequency times time, and it's sine of the frequency times time. Okay, so the idea of FM synthesis is that inside that sine function is another sine function. So you're kind of changing the frequency as a function of time. And that trick turns out, with the different parameters for that frequency changing inside the sine function, that trick ends up giving you lots of different kinds of structures of notes and so on, where you can get a reasonable approximation to a variety of musical instruments. It's not perfect, but it's a first approximation. And the sort of first generation of kind of easy electronic synthesizers, particularly coming out of companies in Japan, was very much based on this FM synthesis idea. Now, subsequently, when it comes to emulating actual musical instruments, this notion of sound fonts has come in, where basically every note, it's like you're saying, play an A. Well, it's like if you say print an A, you say, well, I'm going to print this A, but I'm going to print it with this particular nicely drawn font. Well, similarly, when you make an A as a sound, it's like render that A using the particular sort of structure of amplitude as a function of time, the particular waveform that corresponds to this sound font. And so you might get that sound font by recording somebody actually playing a piano, sampling the sound from somebody actually playing an A on a piano, and you say, that's the sound font that we're going to use. That's the A character, well, it's actually a sound in the sound font that we're going to use for the note A. Things get more tricky because the attack, the kind of how the note is pressed and sort of how the intensity of the sound ramps up in the very early moments as the note is being played. That's important to how we perceive the note, how the decay works, and so on. So it's a little bit trickier, and there's all kinds of detailed technology and tricks to make that sound good. But that's been the more modern way to produce kind of sounds. Now, in terms of electronic music, there's sort of the question of the rendering of the music, the kinds of notes that can get made, the kinds of effects you can get from sort of electronic circuits. There's been not so much done on sort of architecting the structure of the note by algorithms. There's some things done, but it's not been as extensive as one might think. It's not been the case. You know, I know from playing around with our language and Mathematica, we had put in the ability to play an arbitrary waveform back in like 1990, and you could do sort of FM synthesis very easily, and you could start experimenting with different kinds of functions and saying, what do these sound like? Do they sound interesting? Is that like a new musical instrument that you can discover? See, one feature of musical instruments is they have a habit of very much being based on notes. It's, you know, C, D, E, F, whatever, definite notes where there's a definite sort of dominant frequency. And, you know, when it's a string, the reason there's that dominant frequency is the string is vibrating, and there's a certain mode of the string has a certain length, and that means that there's a certain dominant frequency associated with its lowest mode, the lowest vibration of the string. And similarly, when you have a column of air in a winds instrument or something, you're vibrating that column of air, and depending on the length of the column of air, you get a definite frequency of vibration. When you have percussion instruments, it's a little bit more complicated. There's less of a dominant frequency, and there's more of a distribution of frequencies. And I think when you have things like gamelins and so on, you have a much more elaborate, complicated arrangement of frequencies for the quotes notes. But there's a tremendous tendency when one thinks about musical instruments to say, we're going to play certain kind of music on this instrument, and it's specified, the music is specified in terms of notes. And so you want something where there's a dominant frequency and then some sort of color around that frequency, so to speak. And I think less has been done on that might have been. Then there's the question of the actual structure of the music and the way that the notes are the actual you know, tune in the music, so to speak. And there, all kinds of things have been done. There's a bunch of experiments that people have done with, for example, cellular automata, these very simple rules that make complicated patterns that I've studied a lot for the last 40 years or so. Actually, some of the work that I've done there has found its way into composers who are doing kind of algorithmic music. And we've developed things. We developed this thing called Wolfram tones back in 2007, which is a cellular automaton-based music generation system that you can find on the web. The thing that's tended to happen is people sometimes say there's going to be a magic way to make really good algorithmic music, and there isn't quite. Cellular automata do a surprisingly good job at making small snippets of music, you know, 15 seconds, 30 seconds maybe. They have a definite sort of logic, but yet their actual behavior is complicated, so they can make kind of a rich arrangement of sound and so on. And when you render it with the appropriate scales and the appropriate instruments and so on, it can sound quite good, at least for maybe 15 seconds or so. After that, it's kind of like, it's got one idea, and how long can you hammer that one idea, so to speak? Now, people have also tried doing things like pure random walks, or things like fractals and nested patterns. They don't work so well. They don't work as well as cellular automata tend to work. In more recent times, one of the things that's been common, actually some experiments were done on this even earlier, take fragments of known pieces of music and sort of grind them up and reconstitute them in a way through machine learning, for example. Just get pieces of Bach and Mozart or something, grind them up in little pieces, kind of learn the statistics of how those work, and then reconstitute them with something that is sort of statistically like Mozart and see what that produces. And one of the sort of the failure cases there is, well, actually, that cluster of notes is something recognizably from this known Mozart piece. It hasn't been very ground up. It's really just like an assembly from things that have already been done, rather than something where it's learnt the principle and arranged the notes in a separate way. But there's kind of there's a certain kind of idea of having the structure of music be able to be produced algorithmically. And as I say, cellular automata have done a pretty good job on that. And there are no doubt other ideas to be had along those lines. There's also the question of kind of the actual theory of music and to what extent that can be informed by these kinds of ideas and how that affects kind of what is algorithmically possible. So I mean, the current system of 12 semitones in an octave that we have is, I think, dates to, I mean, I think Pythagoras already knew about many of those kinds of things, knew about the idea that there's sort of a factor of 2 difference in frequency between the middle C and the C right above middle C in the next octave, and the fact that there's a major fifth that's a factor of 2 difference in frequency, and so on. And these various musical intervals that get encoded in the positions of notes. I mean, roughly, the frequencies of the 12 successive semitones in an octave go up by 12th roots of 2. So that means each one is just a 12th root of 2 higher in frequency than the previous one. So when you get through 12 semitones, that's a factor of 2 difference in frequency. And there are details of how you actually tune an instrument so that you get nice ratios, like 3/2 ratio of frequencies for a major fifth and so on. And you try and get as many of those as possible. And the 12th root of 2 thing doesn't quite line up with those simple rational numbers. And you have to tweak it a little bit. And there are different systems of tuning that do different kinds of tweaking. And people sometimes experiment with those. And they produce kind of different apparent forms of music and so on. There's also the question of the musical scales, which are the dominant notes that you pick out to make your music out of. And there are 2048 possible ones, given that you've got those 12 semitones and you're deciding which semitones will you include in the scale. And about 200 get used traditionally, between 200 and 400 get used traditionally in kind of the different musical traditions of the world, so to speak. And there's sort of a question of how can you tweak that further. But I would say that there's probably been less done on kind of the algorithmic side of messing around with kind of the actual structure of the notes and the structure of the sound fonts and so on than might have been. I think one of the challenges is that music that is not like anything you've heard before sometimes has a habit of not sounding very good, because some part of the story of lots of these kinds of musical forms and so on is it's a little bit what you're used to is sort of comfortable, and then you can sort of move a little bit away from what you're used to and end up with something that is creative. But if you just sort of throw out a completely random chunk of sound, it's like I don't have any way to sort of grab onto that and make my brain allow that sound to sort of tell a story to me. It just sounds like something incomprehensible, so to speak. And so I think that that's part of that story. And of course, there are sounds that we get familiar with from bird songs and things like this, which then when they are rendered into things like human music, we don't really know to what extent the things that make birds think they're chirping is kind of a good thing, whether that aligns with the reasons we humans think that the music we make is a good thing, or whether there's a sort of connection between what one was used to from the natural world and so on. It's rather unclear how that works. But those are, I think, sort of just going out sort of arbitrarily into musical space will tend to lead to things which are not sufficiently familiar, not sufficiently anchored to, I think, be things that people find terribly pleasing. Let's see. Questions here about, can you build a cellular automaton for each sound font? How does one construct a cellular automaton to map onto functions like sine or waveforms? Well, so what we did in our Wolfram tones experiment is something very simple. A cellular automaton has a line of cells, each one is black or white, and it evolves in a series of steps going kind of down the page. What we did was simply to take that evolution down the page with time, tip it on its side, and treat the cells that are a certain distance away from the midline as being notes a certain distance up on the musical stave. So we're literally taking the sort of black and white squares of the cellular automaton and making them into something that looks just like the early musical notation from 1000 AD of just there's a note represented by this, this square in this musical stave. And then what we were doing was to render that by saying, okay, if there's note number 2 is rendered as a B flat or something with a trumpet or whatever, we're mapping the sort of specific arrangement of positions on this stave onto particular notes and particular instruments. So that was kind of the approach there. Now there's a question, could one make a sound font where the detailed structure, the detailed waveform came from a cellular automaton? And yes, that's a really good question. And a bunch of experiments have been done actually by, for example, students at our summer schools in past years and so on doing that. I would say none of the results have been spectacular, but that doesn't mean one can't get spectacular results. I think one would tend to want to have something where there is probably some built-in continuity to the waveform. You could take something where it's a discrete sort of cellular automaton output and then draw a sine curve through that. That might work, I don't know. They're good things to try. Not as much of that has been tried as one might hope. Let's see. Well, I'm looking at, oh gosh, so many questions about so many different things. Let me see. There's a question here. Oh, maybe I should do this one. There's a comment from WST. Is science slowly becoming a meme, considering how strongly supported the theory has been of natural origin of COVID-19 by the so-called scientific community? It appears a scientist's opinion is more and more becoming a product. You know, this whole story of sort of the mixing of science and politics and public policy and sort of tweaking the public, so to speak, that's happened in the course of this pandemic has been, you know, it's been very frustrating to watch, let's put it that way. Certain things about what's gone on in this pandemic are sort of obvious to anybody who is sort of scientifically oriented, and other things, one might have thought something based on being scientifically oriented and one would have been wrong. And I think one of the challenges is that there's so much that is not understood about immunology, to some extent virology, to a certain extent epidemiology. But I would say the number one place where there's sort of a lack of understanding is immunology, which is a very interesting and important science, but a difficult one that is actually a very computational kind of area, because it's all about kind of the interaction of this whole network of immune system cells and systems and so on. And there isn't a great paradigm for thinking about that. And there are a lot of things which for somebody like me have been like, well, I didn't really know about T-cell based immunity. I sort of was vaguely aware that it was possible, but I thought immunity was associated with antibodies mostly. And I was aware that autoimmune diseases had T-cell components and so on, but I hadn't really internalized the importance of T-cell immunity in certain kinds of viral diseases and so on. And that's just one of a whole array of different things, at least not properly known to somebody like me with just sort of a basic knowledge of immunology. I think this whole question about sort of where did the virus come from? I mean, gosh, you know, my sort of, you know, dominant, very dominant a priori assumption was this was a virus that was the result of a lab accident. And that was my initial, you know, absolute assumption. And, you know, I certainly went and looked up the papers because I was curious about this, you know, produced by the lab in China and so on. And yes, you know, they've been working on very similar kinds of viruses. And it's like, to me, that was sort of an open and shut case, although circumstantial. And, it got a bit more, so we got a bit more kind of in terms of the human operation of the world when I started seeing papers get taken down and things like this. That was like, somebody's trying to cover something up here. So it was, to me, a sort of a just general scientific common sense. It seemed like that was the dominant possible theory. And then when people started talking about, this kind of intermediate animal and that kind of intermediate animal, it's like, okay, show the animal and we'll believe you, but otherwise it's just nonsense. I think that the mechanics by which the scientific community or pieces of the scientific community kind of came in and sort of certified the idea that, it must have been, of animal origin and so on, was pretty unfortunate. And I think that I don't know what kind of reckoning will come from that whole thing, but it certainly was not a great example of kind of free critical thinking being done. And I would say that the efforts on the part of, I don't know, I have to say I will criticize my friends in the tech industry for saying, this is the science consensus, wherever it may have come from. We're going to lock down and lock out every other point of view. I think that was a big mistake. I think that, who knows to what extent that will be seen in sort of the view of history as having been terrible and shocking and the source of lots of problems, and to what extent it will be forgotten, I don't know, but I think it was a big mistake. And I think that it is a challenging thing because one will get both sort of good, sensible science, and we will also get, in a situation of great sort of public and political sort of foment, one will tend to get all kinds of crazy, wacky things that people start to believe. But I have to say that my own belief would be, I might not be correct, that sort of allowing all points of view to be aired is the most likely way to come up with something sensible, rather than saying, oh, no, we already know this isn't true, let's shut it down, which I just don't think is the right approach. I think that that's that has been and I think also there's a great tendency, and it's a very difficult sort of public policy decision. If you think it's important, and it probably is, to get some decent fraction of the population to get vaccinated, for example, then there are things you kind of have to tell people. You might think there are things you have to tell people to persuade them, yes, it really is worth getting this, this vaccine, even if you kind of know that it's sort of scientifically iffy to tell them that. Is that the right thing to do? Is that sort of good for society, even though it might be bad scientific ethics? What's the right thing to do? My own tendency would be to say, just tell people what you know and tell them what you don't know, and try and be as accurate as possible in the communication of the science. And in a sense, it's sort of laziness on the part of the science and science communication to not try and get things clear enough that you can say, here's what we know, here's what we don't know, and to communicate that clearly enough that a decent fraction of people understand it well enough to be able to reason in terms of it and to make the right decisions about, or at least the right decisions for, well, one hopes, and this is where it's again a challenge, is it the right decisions for them as individuals, or is it the right decision for society more generally, based on the things that that they know? But I have to say, I'm, perhaps I just have a view, and the very fact that I'm doing these kinds of live streams, trying to explain things as best I can, is perhaps a sign that I generally believe that it's worth explaining things, and that at least with effort, it's possible to explain some things that might seem to be very much in the scientific stratosphere, so to speak. And yes, it does take effort. I think I find it kind of fun and interesting to put in that effort, but it does take effort. And if I were to sort of replay the pandemic, I would say better science communication would have been a really good thing. And it didn't help that people kind of locked in and said, we know this, we definitively know this, but they didn't. And I must say that I was wrong about some things in my own thinking about what the different paths of what would happen would be. And I have to say, one of the things that affected my a priori assumptions about what was going on was that I was pretty sure this was a virus that leaked from a lab and not a sort of naturally produced virus from the very beginning. And so while I would have had certain assumptions about, certain things are just going to be fine because we've seen N pandemics before, and this is how they always work out, when you see a pandemic that was the result of a essentially genetically engineered virus, then it's like, we don't know what's going to happen because this has never happened before. And so some of the things that it's like, okay, it's just going to work this way, one couldn't necessarily be sure of that. And so that was something which certainly for me affected my sort of a priori assumptions about things. Let's see. There's a question here from Nick. There was and seemed to still be today a very discriminating and political approach in the world of science when somebody tries to present a different way of approaching something. What are my thoughts on how we can improve these sort of strict closed science groups to a broader and more open-minded philosophy? One of the things, one might hope that science would be better if it was bigger, but unfortunately that tends to be a self-defeating thing, because one of the things that tends to happen is the bigger some field of science gets, the more it has to have institutional structure. The more it has institutional structure, the more it tends to want to go in particular directions and to be quite resistant. It's like, well, there's funding that's been set up for doing this and this and this. Oh, you're doing something different. That's not something that this funding has been allocated to. Sorry, can't do that. So it's a funny thing that when science is smaller and there are fewer people doing it, it can be more agile. It's the same thing with, groups at companies and things that, when you have thousands of people doing something, turning that big ship is more difficult than turning something with a smaller number of people. But, I think that this question about what causes there to be the possibility of sort of new approaches, new paradigms for thinking about science, well, a couple of things worth saying. They are almost always the result of one or a small number of people just going off in some new direction. They are very rarely the sort of giant committee of science decides to turn in that direction. That's a very rare phenomenon. Now the question is, how do you get the largest number of people to be these new flowers that are blooming, that are leading to new paradigms and so on? You might say, well, let's have more scientists and then there'll be more possible flowers to bloom. The problem is if you have more scientists, you have more institutional structure, and those flowers tend to get squashed out before they can even get started because there's an institutional structure that's going in a definite direction. But it's the other thing that's always confusing is that when you look at the history of science, 100 years after something has happened, it always seems to happen instantaneously. You don't know that relativity took decades to get decently accepted. You kind of just think, oh, it was introduced in 1905. It was like everybody said, oh yeah, that's the way it must work, and all was good. Didn't happen that way. And in the things I've been involved in, even some sort of paradigm shifts in science that I perhaps been responsible for, it's been a long, grinding, complicated, multi-decade process. And there is a certain tendency for that process to happen only as a new generation of people enter a field. There's a certain tendency for it to be the case that people learn a way of doing things, and that's the way they're going to do things, and that's the way they're going to do it for the rest of their careers. And it's only when a new generation of people come in that it's like, well, actually, we're going to do something somewhat different. And it's always a complicated process, whether the new settlers take over the institutional structure of the old paradigm, or whether they just build their new institutional structure, whether they build a new field, whether they kind of populate an existing field and kind of take it over. And it's happened in different ways in different fields. But this question of how do you get more sort of innovation in science to happen, it's a challenging question. And I think that part of it, I suspect, is you've got to actually, to some extent, bet on people. But that's tough to do in an institutional structure. It's tough to say, oh, the US government is going to put money into this project because somebody believes in this person, even though the project seems kind of wacky based on other things that are sort of part of the established scientific wisdom. I mean, in my own life, I very consciously set things up so that, I'm not asking other people for support for the projects that I do. I have my own kind of structure, and the company that I've run for the last 35 years is sort of the main vehicle that I have for sort of having an environment to do innovative things in science and technology. And that's a very kind of conscious effort to build myself a structure where I can kind of do whatever weird project I sort of want to do that fits within that structure. And I don't kind of have to say, oh, I'm thinking about trying to figure out the fundamental theory of physics based on hypergraph rewriting rules and so on. I don't have to convince some group at the US National Science Foundation that that's a thing that should be funded and that people should give good reviews to it, which I'm sure they wouldn't. I'm sure they'd say, that's a wacky thing. How could it possibly work? It's different from what we already knew. Well, I have to say, back in the day, probably, what was it, 30, 40 years ago, I kind of thought, and to some extent it's true in some areas. So, it's like when you think about scientists, you say, what did these scientists actually do for a living? Well, a reasonable default assumption based on modern times is that random scientist that you've heard of who did some basic science thing back in the 1800s or whatever, I bet that person was a professor, because that's kind of the generic thing that scientists are, is they're professors. Well, I had kind of thought that 40 years ago or something, the generic thing that innovative scientists would be is people who run companies, and that the typical scientist would just have a company. And rather interestingly, that has been in many areas of sort of biomedical kinds of things, computational kinds of things, that has pretty much been the way it's evolved. That your average sort of person who's doing something innovative, there's a pretty good chance they have sort of a company wrapped around the thing they're doing, as opposed to just being a professor sort of operating within the structure of sort of the academic system. So, in a sense, that's a mechanism by which innovative stuff can be done. Now, of course, making a startup company was at one time an unusual innovative thing. And by the time there's a production system for making companies, a kind of, everybody should be an entrepreneur type thing, then that too ends up having an institutional structure where it's like, well, you get your, seed round and your Series A and your whatever, and it's all very structured. And that again puts constraints, and it says, well, you want to do a company that is doing this thing. Well, that doesn't fit in with this type of venture capital funding, so you can't do that. So any time where one of these systems gets big, it will tend to get inflexible. And so that isn't giving a great solution for, it's kind of saying every few decades, shake it up and have a different way to think about the way that innovation is initiated. And I don't have a great solution for this. And I've also seen places where people say, let's institutionalize innovation and just put people in place and have them think for the rest of their lives. That doesn't work out very well either. I think one of the things that can be complicated is that innovation by its nature is a thing that sort of changes over time, and the mechanisms by which it should be done presumably have to evolve a bit, because in a sense, by one method of doing it, it sort of gets stale and stagnates after a little while. So I'm going to need to wrap this up in a few minutes here. There's a question here about from Aaron, sort of thematic. Did summer school and incubator programs exist hundreds of years ago? That's an interesting question. I mean, there was a time when the academics, in the 1200s, when universities started, well, obviously there was a prehistory of universities with things like Plato's Academy, But universities in the Oxford, Bologna, Paris tradition, that kind of started in the 1200s. And you kind of know by looking at the architecture of some of like the colleges in Oxford, for example, with all their medieval walls and ramparts and so on, that it wasn't clear people were entirely keen on those academics, going from place to place in a rather stateless kind of way. And I think this question of sort of who got inducted into, became a student there was very mixed up with the church and with educating people for theology and so on. But I think this question of when did 100 years ago, well, I know that like summer schools and things like physics had sort of come into existence in maybe the 1950s or the 1960s. Possibly there were things before that. I'm aware of sort of ones whose lineage goes back to the 1950s, 1960s. I would say that incubator programs for things like companies, no, absolutely not. I mean, when I started my first company in 1981, it was, you're kind of on your own. There was no sort of institutionalized, this is how to do entrepreneurial things track. And for example, if you look at business schools, for instance, at the time they were teaching sort of, I think, in the General Motors kind of model of sort of professional management, which was there is a company, it is running, this is how you run it, so to speak, rather than this is how to go from nothing to having sort of the entrepreneurial thing started. I think that that's a phenomenon of actually probably the last, the current round of incubators and accelerators in the tech industry probably comes from my friend Paul Graham and his Y Combinator thing. And that started I was in Boston in about 2003, 2004, something like that. I went to some of its very early get-togethers at a converted chocolate factory in Cambridge, Massachusetts. So this question of how do you learn to do business, the answer is, and how do you learn to like start a company? I don't think there was any kind of sort of organized educational way to do that until probably sometime into the first decade of the 2000s. Maybe I'm missing and misremembering something, but when I was starting doing that in 1981, I just don't think there was anything like that. I think it was one of these sort of mysterious areas where you just learn to do it by doing it, so to speak, or you learn to do it in some kind of apprenticeship mode where you work for somebody else's company and then you create your own type thing, which is probably a just fine mechanism and one that commonly happens. Anyway, that's so I think the answer is I at least don't know. There's one more question here. This is bad for me, but from SL, do I think the relationship between the scientific community and media, business, government is fundamentally different today, or pretty close to what we've seen in typical ebb of flow in many decades and centuries in the past? That's an interesting question. I think, I'm not sure that it's fundamentally different. I think in terms of, I'm just sort of rushing into my memory are all kinds of examples from the 1800s of kind of science meets government. Like, for example, the famous example of Faraday showing Gladstone, who was then prime minister in England, some electricity thing. And, Gladstone asks, okay, Faraday, what's this good for? And Faraday is explaining, like, it's like a newborn baby. It's not clear what it's going to turn into and so on. And apparently Gladstone's eventual sort of question was, well, okay, but can we tax it? What is the relationship? If this is going to be a useful thing, how does it mesh with the government? Is it something that the government can kind of control? And that's a story that's been told and retold over and over again. That's not really anything new. And the kind of the enmeshing of big progress in science with military needs and other government priorities, that's a story that's been being told for a couple of thousand years at least. I think that when it comes to kind of the world of business, it's there have been times when academia has felt itself to be this very ivory tower kind of thing that was sort of at cross purposes and a different vocation from kind of the business world. And I certainly, in the early 1980s, for example, that's certainly what I saw, was people were like, I'm an academic and I'm proud of it, and I'm going to live in an ivory tower, and I don't want to have anything to do with that icky business world out there. And business people were like, oh, they're these academics and they had their heads in the clouds, and occasionally we might hire them as a consultant, but it's kind of like they're off doing their in the clouds type stuff. Now, obviously, there were continually cross connections, and there were continually companies being started that had sort of come out of academic research. And, you know, people like Edison, you know, he was certainly fond of reading the literature and pulling in professors, and, you know, Howard Hughes, the same kind of thing. Lots of people sort of pulled in from academia to help with essentially industrial research and so on. And then there were the occasional scientists like William Thomson, later Lord Kelvin, was much involved, that was in the 1800s, was much involved in, for example, the laying of the first transatlantic cable. You know, he was famously, the cables kept on breaking. And so Kelvin was there on the ship, so to speak, eventually with an electrometer kind of listening for the resistivity of the cable to try and predict when it would, you know, when they should slow down, whatever else it was. And I think he was involved in the first successful laying of a transatlantic cable that didn't break. And, you know, that was one of those typical investment things of the time is, you know, you'll invest in the transatlantic cable. If it gets laid successfully, you're a big winner. If it breaks somewhere, you know, on the mid-Atlantic ridge or something, then you're toast, your investment is sort of buried under the ocean type thing. So I think there were certainly individual people who were involved in, I know Kelvin did quite well from his stake in whatever company it was that eventually laid that transatlantic cable. So yeah, I mean, I think that a lot of these things have been happening. I think that as academia got bigger in the 1960s and 1970s and so on, there was a certain sort of, you know, separation of vocations that happened, which I think has tended to be more merged in the present day. Although you still see sort of echoes of the, you know, we're academics and we don't want to have anything to do with business, and we're business people and those are just academics and they're doing crazy, useless things. The role of media is interesting. I mean, I think that the newspapers had covered sort of scientific advances, although seemingly in a very kind of down-to-earth way for a long time. I mean, you know, Einstein was a big celebrity when he came to the US. And but even I remember like the headline from the New York Times in 1919, when Eddington did this expedition to South Africa, which claimed to validate Einstein's general theory of relativity. You know, the headline in the New York Times was something like "Lights Askew in the Heavens." So it's definitely not, you know, not science terminology. It's kind of bringing science terminology to something that sounded more kind of for the everyday person. And I think the life and times of, for example, science journalism have been interesting to watch. I think the 1980s may have been the peak of when, particularly in the US, science journalism was at its most vigorous, with, you know, every significant newspaper having its own, you know, science columns and things like this, and a bunch of science magazines getting started and so on. And that didn't really work out in the US. It's worked out a little bit more in places like the UK, you know, like New Scientist magazine, which I realized recently I have had an unbroken subscription to New Scientist magazine for 53 years. The only magazine where I've had such a subscription. It's a magazine that you know, it has its ups and downs, and it has a certain degree of British humor, which might not be, which I still find entertaining. But it's been, in the US, the sort of science journalism world has not been so successful. I don't know exactly why, So, you know, that, but now when it comes to things like the pandemic and the interaction of media, it's been, in my opinion, extremely unimpressive. I mean, people in, you know, the mainstream large scale media outlets, you know, it's like, come on, guys, you know, did you do no math class? Do you really not understand how to divide these numbers to, you know, come to a conclusion? Are you really, you know, are you really that clueless? I've been surprised at how generally poor the coverage of these things has been, particularly when it comes to more quantitative issues. And it's been surprising to me that journalists, for the most part, have not been prepared to, the mainstream journalists, not bloggers and people like that, but the mainstream journalists have been so unprepared to actually do any rolling up of their sleeves and like, let's try and figure out, what does this actually mean? What do these numbers, you know, how do these numbers actually fit together? There's been a kind of a, no, we can't do that. We're just journalists. We're just reporting what people say type thing, which has sort of surprised me. And I don't think that would have been the case at different times in history, but I'm not sure what, and I have to say, I think right now, I will say, and probably I'll offend people by saying this, but I think mainstream science journalism is quite weak right now. I think that, for example, you know, I notice with our physics project, it's, you know, it's covered in lots of places, but it's definitely, if I say, should it be in this mainstream, you know, this place science coverage, that piece of science coverage, it's like, well, I don't know. It's not the kind of thing these guys are writing about. They're writing about much more mundane things. This is a much more complicated thing to understand. It takes much more effort. It's really not the kind of thing, and the kinds of things that are being written about are much more, frankly, much more straightforward and require much less effort on the part of the science journalist. You know, why it's evolved that way, I'm not really sure. I think it's a shame. And perhaps it's related to my previous complaint, so to speak, about people's unwillingness to try to actually explain science to people at large. And in a sense, I'm trying to do my small part here with these live streams to try and explain some things that I know something about in a way that I hope people can understand and relate to. And you know, more people should do this stuff. Anyway, unfortunately, I have to wrap this up here. And as I said, my next few weeks are very dominated by these educational activities that we have every summer. And probably this particular series about history of science and technology is probably going to be on hiatus for the next 4 weeks, although we will be doing a variety of live streams, which may include some about history of science and technology as part of our summer school and summer camp. And maybe somebody knows that schedule. I'm afraid I don't yet know it. But look forward to people joining us for some of those live streams. And I hope you found this interesting, and I find it interesting to try and explain things, and I look forward to seeing you all another time. All right, bye for now.